All right. So today I will talk to you about the relationship between citric acid exudation by roots and phosphorus desorption, and how is that related to the disaggregation of soil colloids. This is a field flow fractionation study, which is a neat technique that I will explain in a bit. And our main finding was really showing the link between this phosphorus desorption from iron and aluminum particles. And this is as a direct response to the presence of citric acid on the soil solution. So first, let's explain a little bit about the background. Why phosphorus? Yeah, this is part of the project, the big project that we had on UK called Opus project. I will give a link for you here about this project. And this project was about plant utilization of phosphorus from organic matter in the soil, so organic phosphorus. Organic phosphorus is not directly available for plants. It needs to be first mineralized by soil enzymes. In our case, we were hypothesizing in this project that we needed a two-step process. One is the solubilization of the organic phosphorus, and that involving organic acids exudated by plants. And a second step would be the mineralization through enzymes released by both plants and microbes. And this work is related to that solubilization of phosphorus and desorption of phosphorus caused by citric acid, which is the main organic acid released by plants into the rhizosphere. And it has been demonstrated that this exudation of citric acid is linked to a higher uptake of phosphorus by plants. So the higher the exudation of citric acid, the more phosphorus uptake by plants. And even there are some works that are using this as a biotechnology, meaning that plants are being engineered to release and exudate more citric acid into the soil solution. And by doing that, they can mobilize more phosphorus where phosphorus is recalcitrant and unavailable for plants to utilize. Yeah. So, in this work, it's a follow-up work in a previous work that we've done, a DGT work. The previous work, we showed the relationship between the desorption kinetic parameters and citric acid exudation in the rhizosphere. But from that work, we saw that there was a significant change on the rate constants of the kinetic parameters from phosphorus desorption, and we didn't see why that should be happening. So one of the sub-hypotheses that we had from the previous work is that these changes on phosphorus desorption kinetics were due to the changes on the exposure of the surface of the soil colloids. So, namely saying this, the soil colloids, they are forming aggregates and microaggregates, and these aggregates, they have occluded phosphorus. Some phosphorus is available for desorption, and some other phosphorus are hidden from the soil solution because they are inside these microaggregates. So in order to demonstrate that, we should show that by applying the citric acid in the soil, we are showing that this disaggregation is happening, and this aggregation is proportional and concomitant to the appearance of phosphorus in the soil solution, and also to the changes in the soil the phosphorus desorption kinetic parameters. So this is what we've done in this study. We have this hypothesis that this disaggregation was actually what was happening, and we went out to measure the appearance of these fine colloids, water-dispersible fine colloids, and at the same time correlate to the appearance of the soluble phosphorus and to the changes in phosphorus desorption kinetic parameters. So let's talk a little bit about the methods. This is, as I told before, is a field flow fractionation study. Field flow fractionation, or short FFF. FFF is a technique where we have we apply a field across the perpendicular to the flow of the solutes, and by applying that field, in this case a hydraulic field, it allows for the separation of different sizes of particles. Here you will see a video, and this video shows that how it works, this method. Actually, you have the flow is happening across a membrane, a PAS membrane of molecular cut of 1 kilodalton, which is a little bit below 1 nanometer size. So it means that through this membrane, all soluble phosphorus and any ion soluble in the solution can pass through, but not the particulate matter that is suspended in the water. And by having this flow perpendicular to the current, what will happen is that it will start separating the size of the particles. And the finer the colloids are, the quicker they will go through this process, and the bigger the colloids are, the slower they will move through this fractionation procedure. And at the end of this, we have a UV visible detector, and also we will have ICP-MS, which is hyphenated with this system. ICP-MS will be doing a total elemental analysis on this. So we can see if each one of the eluted fraction is what is the composition of this fraction. So we can see, for example, how much iron, aluminum, phosphorus, carbon, manganese, and silica there is present on these colloids. And by doing this, we can see if they're different from each other first, and what is the size that it corresponds, what are the size fractions. And we can, by changing the dose of the citric acid and changing the time of elution, we can make these correlations and see what is happening, what is the response pattern that we can see from the exudation of citric acid and the appearance of these different fine colloids. We can do the same and analyze in the total concentration of these same elements: phosphorus, silica, iron, aluminum, manganese, and carbon. And by comparing this, we can see how much was soluble, how much was present in this colloidal fraction. Another thing that I need to mention is that how do we know the correlation between these, the particles, or the size of the particles that are being eluted on the field flow fractionation? We run first latex standards, sulfate latex standards, and these sulfate latex standards will give us peaks, as you see here on this animation. And these peaks, we can assume that each one of these peaks corresponds to a particle size, and then we can assign those fractions to what we observe when eluting the fractions from the soil colloids also. So let's go first for the results. There's another thing I have not mentioned. We did this fractionation by filtering the soil suspensions through different syringe filters. So one syringe filter was 0.22 micrometers, and the other one was 5 micrometers. And we found that the 0.2 micrometers give us a lot better resolution for the majority of the elements, except for silica. Silica was, in most of our extracts, we find very little silica colloids when filtered to below 0.22 micrometers. But when we did filter to below 0.5 micrometers, yes, we found the good patterns of response for silica. So actually we use the 0.5 syringe filter only for the silica fractions, but we have not used it for the phosphorus, aluminum, manganese, and carbon fractions. Okay, so another thing that I must say is that with these we didn't have a very good resolution for silica, but we also did not have a very good resolution for manganese. It was a widespread peak, so we kind of didn't use manganese at all in this study. We just didn't use the data because it was very strange data. And the dilution time showed us, first finding that we have is the dilution time that we obtained showed a very good alignment between the peaks of phosphorus and carbon, in an interesting similar behavior between the dilution of phosphorus and carbon through both the time of elution, time of incubation, and also the dose of citric acid. So by observing that and by seeing also that these peaks were offset from the peaks of iron and aluminum, the peaks of iron and aluminum were also aligned with each other, but the peaks of carbon and phosphorus were offset from iron and aluminum. And that was showing that for us, that is a good evidence that the type of phosphorus that we are observing, that is this nanoparticulate phosphorus that is about a little bit below 20 nanometers, that is organic phosphorus, actually. The response of this iron and aluminum was similar to each other, and the response of carbon and phosphorus were related to each other. So this is what we're going to show now. So what we find here is that here you will see both the graphics showing the total concentration of phosphorus and carbon in these extracts. Let's show first phosphorus, and then let's show carbon. Yeah. So the total concentration of phosphorus in time, we have incubation times of 0 hours of incubation, 2 hours of incubation, 12 hours of incubation, and then 24 hours of incubation. What we find is that the response of the phosphorus did not change too much with citric acid in time. So there was not a huge increase in colloidal fraction carrying phosphorus in time. And if we look at those also, we will see there is a slight increase in phosphorus with increasing dose of citric acid, but they were not statistically significant. So let's say that here there was not a significant increase in phosphorus in phosphorus nanoparticles, in phosphorus colloids that were being mobilized by citric acid release on the soil. So there is not a huge amount of nanoparticles carrying or water-soluble colloids carrying phosphorus in response to the release of citric acid. In carbon, nonetheless, the increase was not so high, but yes, it was significant at both in time. Here you will see at 12 hours and 24 hours, there is an increase in the amount of carbon being released. There's more organic matter colloids appearing in response to the citric acid. In time, with the longer the incubation, the more you see it. And also with increasing doses of citric acid, you see a significant increase in the appearance of more carbon. But this response is not very, although it's significant for carbon, the amount is not so high at all. Now let's talk about iron and aluminum. So iron and aluminum, as you see here, there is the same animation that we make for carbon and phosphorus. First, in time, you will see that there is a significant increase, nearly doubling the amount of iron and aluminum present in colloidal fraction with the increase in incubation time from 0, 2 hours, 12 hours, and 24 hours. So it increases significantly the amount of iron and aluminum in a similar pattern. And if we look here about the response, about the dose response of this, also you will also see that the more than 10-fold increase in iron and aluminum present in the colloidal fraction measured by this FFF coupled with ICP-MS technique. Yeah. Silica, on the other hand, we, as I told before, we didn't see a lot of a good pattern with the fraction of below 0.22 micrometers. So I will just show you here that what we were able to measure this to below 5 micrometers, and there is a much higher molecular weight fraction that you can see here in silica. So also we measure the total concentration by using the same methods, and this total concentration, we can compare this pattern of the total concentration to what we find the concentration in the colloidal fraction, and then we can see how much of it was soluble. And we see that this response was very similar to what we saw in the previous study, using where we measured the phosphorus desorption parameters, where we have a slight increase, nearly doubling the increase in phosphorus, nearly doubling the increase in carbon, and a very significant increase in iron, aluminum, and silica soluble in this system. Although silica, you do not see that in the colloidal fraction, you do see it very strongly on the soluble silica in this system. We, of course, use this data together with the phosphorus desorption parameters and the colloidal data, the total concentration data, and the phosphorus desorption parameters, and we established a correlation patterns where we've seen that we could observe and make the link between the desorption of the disaggregation, the appearance of fine colloids from iron and aluminum, highly correlated with the desorption kinetic parameters, meaning that the disaggregation of this iron and aluminum is leading to the change of the kinetic of the phosphorus desorption in soils. And the ozone phosphorus, which is usually used as a bioavailability measurement of phosphorus, the ozone phosphorus was only correlated with the disaggregation of silica colloids, and which could suggest, this is only one soil sample, but it could suggest that ozone phosphorus is more related to phosphorus which is adsorbed and occluded on silica particles and silica colloids, whereas the DGT measure desorption could be explaining the more of the phosphorus which is coming from the iron and aluminum associated phosphorus in the soils. Yeah. So just to conclude very quickly, we have shown our findings, shown that there is in this soil specifically the phosphorus, the nanoparticulate phosphorus, is organic, and there is a slight response of this to citric acid, but not very high. It's a water-soluble phosphorus, but this desorption of this phosphorus could mean that it's being made available for enzymes to hydrolyze this organic phosphorus. And then we have shown also that the origin for this situation, for this case study, the origin of the desorbed phosphorus is coming, the majority of it is coming from iron and aluminum colloids, desorbed from iron and aluminum colloids. And some of it is coming from silica, but not so much from silica as from iron and aluminum colloids. Okay, so this is all what I had to bring for you today. I hope you enjoyed, and thank you very much.